Now, mutations can be permanent and they can be inherited. Most of them are harmful, but sometimes they give you some adaptive or evolutionary advantage. There's the Ames test that screens for any carcinogenic chemicals, because if it can mutate bacterial DNA, it probably can mutate mammalian DNA. Recombination is when a bacterium donates DNA to another bacterium, and that causes a new strain that's different from both the donor and the recipient. And this is bacterial plasmids and gene exchange. A recombinant organism contains and expresses genes that came from another organism. Genes can also be transported horizontally, and this usually involves small pieces of DNA, like plasmids or chromosomal fragments, and these plasmids replicate independently of the bacterial chromosome. And then these chromosomal fragments will integrate themselves into the bacterial chromosome so they can replicate. And there's 3 ways for this to happen: conjugation, transformation, and transduction. With conjugation, it is where you're going to have a donor cell with a pilus or a pilus that will transfer DNA from the donor to the recipient. And this, an example of this would be drug resistance. Transformation is where you've got the donor DNA in a fragment or a plasmid, and the donor is usually dead, but the recipient is not. And transduction, the donor is lysed by the bacterial stage or cell, and then the defective bacteriophage is the carrier of the donor DNA. But the recipient has to be the same species as the donor.